Okay, everyone, welcome to this week's seminar. I'm really pleased to introduce Dr. Brendan Pinto, who has come here to ASU through the route of first Morningside College in Iowa, then to Marquette for his graduate education, and then a brief stop in Texas for postdoc. And then here for postdoc, and he is still with us, although technically working for the NIH, but we get to see him still. So please join me in welcoming Brendan today.
And start. Awesome. Okay. So today I'm here to talk to you about sex chromosomes, Gila monsters, SCINK, but not like this. We're gonna do something else. I gave this talk title a while ago, so we're gonna flip it. Monster, SCINK, and initial insights from the first complete squamate genome. I'm very excited to be here today to talk to you about it because I finished it last week. So this is like hot off the presses. So let's get to it. First, cast and crew. I want to give a shout out to our postdoc Simone, who's done a lot of the large scale analysis for the first part of the talk. Dale, obviously, who's here because I'll be talking about the Gila monster, and we would not be doing Gila monster work without him. And then Melissa, obviously, for obvious reasons that I'm not going to belabor. And then I also just want to give a shout out, and also this is a precursor to the end of the talk as well, to all of the people also working in this field who make my work sort of possible. So here's our roadmap for today's talk. I'm very convinced that telling you what I'm going to tell you, and then telling it to you, and then telling you what I told you is the right way to give a talk. So here's what I'm going to tell you about. We're going to start with background about sex chromosomes in lizards. We're going to talk about ways to identify sex chromosomes across diverse taxa, and then we're going to get into the Gila monster towards the end of the talk. Sex chromosomes. So sexually reproducing animals need some mechanism in which to bend some individuals as egg havers and some as sperm for sexual reproduction to work at all. And there's a ton of different ways to do this. One of which, and one of the most familiar with, is a genetic mechanism, male heterogameity, where the males possess a sex-limited chromosome and females possess two X's. You can have the inverse relationship, where females have the sex-limited chromosome and males are the homogametic sex. And then you can have totally other things like temperature dependence, where all the chromosomes are autosomes, and you don't have to worry about any weird stuff happening. But sex chromosomes come from autosomes, which makes them interesting and difficult to work with. Where one of the two autosomal pairs acquires a sex-determining locus, or an allele, in which this is generally a dominant genotype that influences the either development of testes or ovaries. And over time, through a variety of mechanisms, recombination can be restricted around this region. And what that leads to is what we call degeneration, where the sex-limited chromosome, the one over on the right side of the graph, accumulates mutations, loses functional genes, and accumulates repetitive elements. And over time, similarly with as we know in mammals and birds, we have sex chromosomes look like this a lot. That being said, most vertebrates possess sex chromosomes that are somewhere in this stage up here. So under the light microscope, they look completely identical, and we need other mechanisms to identify them. So in other words, these are easy mode, these are hard mode. So let's get into the taxa that do the hard mode, which are the lizards and snakes, or one of these examples. And I need to emphasize very quickly that This is the most successful vertebrate clade on Earth. There's over 12,000 species of them. They are older than most other groups. They have lovely phenotypes, which I'll get into today. But birds are often considered to be the most speciose. They are not. Mammals aren't even close. There's a lot of cool things going on in lizards. So I just want to emphasize this point towards the beginning of the talk. And just for scale, this line right here, just about 200 million years ago, this is the line where extant mammals diverged. So this would be the split between platypus and human, is right here at about 200 million years. And so that's when squamates also evolved. That is the basal, most recent common ancestor of squamates. And here, these pink and orange lines are when birds and mammals, the placental mammals and all birds began to diversify, which means that they are extraordinarily evolutionarily young relative to squamates. So squamates have been on the Earth a very long time, and they've been very successful, and they're very interesting. They've evolved things like being the smallest amniote, like the spherodactylus lizard here, and some of the largest, like the mosasaur, which is obviously now extinct, but it is not a dinosaur. I'm wishing. Better? Hopefully. But just because they're the most successful vertebrate clade does not mean that they are the most recognized as successful or invested in as successful. And so if we look at numbers of published genomes that are out there on NCBI or whatever, this is a figure from Darren Card's work. But as you can see, throughout the years, mammals got a significant head start as far as vertebrates go. I guess this is amniotes only. But birds and mammals have a lot of genomes. So this is percent of relative species abundance. And then this is total number of genomes. So they're doing pretty good, relatively speaking. And although this plot ends in 2022 and the numbers have changed, the trend has remained. And so you have to actually zoom in to see the squamates, where it's still less than 1% of all species have had their genome sequenced. And so there's work to be done. And the interesting part is that it doesn't seem like anything major is changing. So I can tell you that they're the most successful vertebrate clade, but somewhere like the Vertebrate Genomes Project, who is actively trying to sequence every vertebrate, has systematically unsequenced squamates. So lepidosaurs are squamates in the Tuatara. Here's the progress of, so green would be genomes finished by the VGP, and orange would be genomes unsequenced by the VGP. And although birds, fishes, even sharks and rays are actually doing rather well, the squamates remain unsampled. So if we're going to do cool work on this ancient clade, we're going to have to do some of the heavy lifting ourselves. All right, sex chromosomes. Sex chromosome identification. The lizards and snakes are a great model for sex chromosome evolution. It's because they do a little bit of everything and then some. So we have ZW listed here in orange, geckos, laceridids, angelmorphs, acrodonts, snakes, pleurodonts, XY, which is the blue, geckos again, skinks, teiids. There's a large diversity of what things are doing. And emphatically, just because the tips are all labeled as orange or blue doesn't mean that it's the same orange or blue, in that the genomic location of the sex chromosomes can change between taxa. Can and does. So for instance, if we look at our angelmorphs, which is going to be like a group of, that's the rug that's going to hold the talk together today. It's going to bring everything together. We're going to look at the angelmorphs right here. It's the least speciose group, but these guys tend to be some of the most charismatic. They tend to be large-bodied and long-lived. Things like the Komodo dragon, the Gila monster, crocodile lizards, alligator lizards. These things tend to be pretty interesting to study. They also, all recorded species have a ZW system, although we know it's not all the same ZW system. So that's interesting, and I'll pick that apart a little bit as we go through the talk. And so for scale, our angelmorphs are here in this young sex chromosome, the ZW, but generally looks similar under the light microscope. As an example, I want to run you through the difficulty in identifying a lot of sex chromosome systems. So this is an example from the crocodile lizard, Shinosaurus crocodilus. This is a monotypic family of endangered things, and so identifying more about its biology might help us be able to conserve it. And so what we did is we simply took a large data set that was available after we tried our own experiments and failed time and time again. We tried somebody else's data, and we found that there is a ZW system. It's not similar to any other ZW system that we know in the group, and it's located here at the end of chromosome 3, or linkage group 3, I should say. And this is cool. This is cool, but, oh, hold on. This took 50 individuals to find this one tiny little locus. We did a lot of bioinformatic-y things. We invented a technique that was just sort of, I don't know, made sense, I guess. We took a bunch of resequencing data and we pooled it together. Normally, you do this thing called pool-seq, where you sequence one pool of a bunch of individuals, and we just did this in silico because we didn't have a pool-seq data set, and it seems to have worked. And I'm just going to put a pin in this, and we'll come back to it later. But now we need to talk about a better way to identify sex chromosomes, which is SCINK. And so this is going to be the main chunk of the middle half of my talk, is who, what, where, when, why, and how. So to identify sex chromosomes, we need to understand what their composition is in the genome. And although, again, any linkage group can be a sex chromosome linkage group, any gene can be a master sex-determining gene or a primary sex-determining gene. But there are things about sex chromosomes that are consistent across taxa. So one of which is that chromosome length is strongly correlated with how many genes are on the chromosome, which is intuitive. GC content is also correlated with the length of the chromosome. And one thing that we were particularly interested in is that genetic variants are also correlated. So the number of SNPs you have is very strongly correlated with how long the chromosome is, which makes sense because the number of base pairs is also very strongly correlated with how long a chromosome is. And this is very true in humans, for instance. So this is interesting, again, because humans don't have microchromosomes, and so you can see the steady stepwise decline between some of the smaller chromosomes and the largest. And although it's not, the straightest line you've ever seen, it's obvious that there's a relationship there. And so this presents us with an opportunity because genomics has changed in the past couple years. We can now assemble not only the genome as a reference, but we can assemble mom's haplotypes and dad's haplotypes separately from the same individual, and we can combine them, and so this is what we call the graph. And so red would be, say, mom's haplotype, and blue would be dad's, and you could do this for every single chromosome. And what we then have is effectively, we sequence 2 individuals for the price of 1, which could be useful if used appropriately, or intuitively, I guess. And so if we know that genetic variants are correlated with chromosome size across sort of at the population level, if that's true within an individual, we can use that to our advantage. So if we were to turn our chromosome level assemblies from mom and dad into a bunch of k-mers across the whole genome, so this is an autosome, here's our sex chromosome system. If we subtract the number of k-mers, or the words that make up the chromosomes from each other, there's going to be some amount of background noise, or SNPs, that's correlated with genome size or chromosome size, like I mentioned. But in the sex chromosome specifically, there's going to be one region that's non-recombining that has an excess of these k-mers. And that is going to give us this correlation, and it's going to give us some statistical backbone to compare against as well. So real quick, I want to jump back to a squamate. This is one of the first, actually this is the first hi-fi assembly I did for PacBio. This is the leopard gecko. But what's important about the leopard gecko is it does not have sex chromosomes. So this is a temperature-dependent species, which means that all 18 of its chromosomes are going, or 19, are going to be autosomes. So whatever's going on here is going to give us a strong indication of what things look like without sex chromosomes. And we see an extremely strong correlation at the autosomal level. So an R squared of 0.9, which is really high. And so this gives us a reasonable understanding that, sorry, I don't have the thing right here, so I'm going to be flipping back and forth to make sure I know what's happening. But yeah, so this is consistent with this genetic variation is dependent on chromosome length just within a single individual. And so what this also gives us is sort of a, for lack of a better term, a statistical trend that we can then test against. And so I'm just going to start adding sex chromosome systems to this plot as we go through them. It's going to be kind of rapid fire, but I'll start us off slow. So again, we have our autosomal trend, and we have some amount of statistical wiggle room to work under. So if you turn that plot on its side, you would see something like a normal distribution. And so I'm going to show a lot of these. First and foremost, we have our dot plots. This is the plots that I've just been showing you. Occasionally there will be a bar graph here in the corner, but on the right side is going to be one of the sex chromosomes aligned to the other. In this case, it's a ZZ, so this is a male wall lizard. This is going to be one of the Z chromosome haplotypes, and this is going to be the other one. And it just goes, this is just going to run through sort of rapid fire, but this is going to be the same plot a bunch of times in a bunch of different species. So for this one, we're going to see our two Zs. These guys look just like autosomes, so that's where they're going to show up on our plot. Another option, we're going to go back to the crocodile lizard. So this is data shared with me by Hung-Shin Zi at the University of Glasgow, who recently they finished the haplotype resolved assembly of the crocodile lizard, and he sent me a plot. He's like, I used your program, here you go. And I said, cool. I don't see the sex chromosomes because, as we know from the previous slide, it's just a very tiny region, and there's almost no possible way that that could generate a signal that would be visible at this scale. But what that tells us is that most vertebrate systems, if you sequence the homomorphic sex, you're going to have your sex chromosomes falling on the trend line. And in undifferentiated homomorphic systems like the crocodile lizard, again, you're still not going to see anything. So there are limitations here of what you're going to find, but now we can talk about the more interesting cases in which you would sequence a heteromorphic individual that has detectably interesting sex chromosomes. And so, for instance, if we look in a human or a mammal or a chimpanzee or whatever, we're going to see this highly degenerated Y chromosome and the X. Again, here's a pseudoautosomal region, which is the only part that really aligns. But again, we get our strong correlation between our haplotypes, and then the X and Y fall way off the trend line. In fact, you can't even really see them. We cut them off this plot because it looks terrible. I mean, look, that doesn't look like a trend at all. But they're up there somewhere. Again, these are the unique bits that give the signal, and so the more unique, obviously these look very unique from each other, the stronger the signal is. So we can do that in a bird, like this parakeet here. So again, we have a really strong correlation, and here's our ZW way off the chart somewhere up north. And then we have a wall lizard that also has a ZW. This one's, I would say, less differentiated in that the W is not as small as birds or mammals. But again, you see this ZW falling way off the trend line, but we still have our strong correlation. So from this, we can say that extremely heteromorphic systems are going to fall way off the trend line somewhere. And in a moment, I guess I will show you that homomorphic systems will also fall off the trend line. But at a more interesting level, what happens when you have like an XX for a mammal? And since we know that the X chromosome in mammals harbors less genetic diversity than all the autosomes, we can actually see that here. So if you sequence a mammal of an unknown origin and you sequence its genome, you can tell whether it's XX or XY, because the X's are here in an XX individual, and the XY would be way up here somewhere in an XY individual. So this is the case in the red fox. And again, we know this data from humans that it's because the chromosome X is down here. And if you did some statistics on the two chromosomes that are closest in length there's significantly less variation on the X than autosomes. So, and that tends to be true across mammals. This is also true to some degree in birds, where the Z's tend to be lower than our autosomal trend, but not statistically significant or whatever in that context. So yeah, there's one last part of the plot here, where if you have extremely ancient systems where repeated bottlenecks have reduced genetic diversity on the X's or the Z's, you can actually see them on the plot, which is really cool. Because again, most species, that's not going to happen. So that's a mammal thing. A lot of things are mammal things. But again, we have our sort of normal distribution, and at some level, we can have our sex chromosomes falling off to above the trend line, or we can have X's and Z's falling off below the trend line. And either way, we can identify them just from the assembly without having to know what they are or where they came from. So does this work in practice? Can we use this across a bunch of vertebrates? The answer is obviously yes, or I wouldn't be here talking to you about it. But we're going to go back to our squamates, because these guys are doing a little bit of everything and more. So we'll start with a couple skinkomorphs. So first, this is Xanthussia vigilis. This occurs in Arizona, and it was collected in Arizona, not too far from here, with the help of Levi Gray. And what we found is that they possess a ZW system, although it's not the same ZW system as their closest relatives that we know have a ZW, and so, of course, more complicated than it needs to be. And then for just this plot, I've changed what's on top to what's on bottom. So Levi and I collected 4 or 5 individuals of these guys, so we have population level data, and that's what this top plot is. So this is female over male FST, and so we see this clear sex-limited pattern right here, where high genetic divergence in this region would suggest that it's a sex chromosome. We see the mirror image of that just within the SCINK results in the mirror plot here. And so for the most of these, I'm going to have one of these bar things on top and one on the bottom. But for this one specifically, I wanted to emphasize that this took 4 individuals to identify, and this took 1 individual. So obligatory meme, look what they need to mimic a fraction of our power. So now that we've done skinkomorphs, we can do a snake. And obviously all snakes are this orange ZW, even though all snakes are not this orange ZW. And I can show this because this python here is an XY system. And so this is data generated by Sarah Carey at Hudson Alpha. And again, we just see these strong patterns and strong correlations that we can use to our advantage. So I'm just going to rapid fire through a couple more now. So we can use this in geckos. We can identify complex systems. So in Certipodian scabrum, which has been introduced here in Arizona and was also collected here in Arizona, we have a W autosome fusion creating this neosex chromosome, and then we have the 2 Z's that align with that W during meiosis. And when we align them, we can see this region with really high Kmer densities or hapmer densities, and this region of reduced percent identity. So red is going to be more identical, blue is going to be less. And so again, this fits our mold of what a sex chromosome should be doing, where this side is a par region, and then this side is also a par region. We can click through a few more. So this is a skink. This is actually an extinct skink in the wild, but it has its genome sequence, so we can use it. Again, we know that it has an XY, and we know the linkage group, and we can find it from just a single individual. Hemichordilus, a South African skinkomorph, has an XY system. We can identify it rather easily because it turns out it's heteromorphic. We can do this in something super divergent like a ray or a shark. And again, we see the XY system falling way off the trend line, and we still see a strong correlation just within the autosomes. So things are working, which is cool. And then lastly, we can do this for another anglomorph, because we always are going to pull it back to anglomorph somehow. And Aniella stebbensii, this is a San Diego legless lizard, has a ZW. This has never been reported before, but Again, it's an anglomorph with a ZW, which is a shocker, except for, again, this is a new ZW system that we've never seen before. So we have a mix of being able to validate things we already knew, and then because of that, we think we have what it takes to start identifying new things just based on these kind of results. So I just showed you that we can use SCINK to identify X, Y, and ZWs in skinks. We can use it in laceridids, use it in anglomorphs, snakes, and geckos, and other things too, but these are just the ones that I showed you. So it's super effective across a breadth of diversity, and that's really cool. One individual, let's go. Okay, I'm gonna stop briefly and ask if anyone has any questions because we're gonna totally change topics here now. Totally, mostly.
For the snakes with the XY and the ball python there, sorry. What proportion of snakes you looked at show an XY versus the ZW?
So the originally thought ZW system and all snakes share is only evolved and is retained in advanced snakes. So colubrids, vipers, elapids, and their relatives. So all of the earlier primitive snakes, I'm using air quotes if you can't see me. So pythons, boas, blind snakes, all of those, they never had that ZW system. And so it turns out they're the Wild West, just like geckos are, or acrodonts. So they have XY, ZW, and other things too. Secret third options. There's a triploid blind snake we don't know what's going on there. But yeah, it turns out that the thing we always thought that we knew, we didn't ever actually know. So, cool. All right. I can just move it. Yeah.
That's kind of stuff I know. What is the problem with the whooshing sound, or?
The scratching.
Yeah, so the only way we can do that is by pulling your shirt out and pinching right to it.
Okay. Oh, good. Awesome. Okay. More anglomorphs. This one's a little more iconic than the last. Which is, it's going to be the Gila monster, but first I want to stress how cool this is by giving you a snapshot of genomics through the eyes of the NHGRI. So the Human Genome Project obviously happened about 10 years period from 1990 to 2000-ish, and it concluded in 2003, and that was housed originally in the NHGRI. In 2011 was the first squamate genome published. This was the green anole, and this was published in coordination also with the NHGRI. It wasn't until 7 years later that the first chromosome level squamate genome, which was a python, like I just showed, was put out into the world. But the human genome was finished 4 years later, Chem 13, which for those of you who are, and this will only come up later when I tell you how I still have one chromosome I haven't finished. When they finished the human genome, there was no Y chromosome in it. So I just want to pin that really quick as a preemptive justification for what I'm going to say later. And then just last week, September 18th, the first complete squamate genome was finished. And I did that. That's cool. So, and again, because technically we're at the NHGRI now, then we get more NHGRI stuff in here. So that's it. First complete. Let's talk about it. So the first, it's the Gila monster. It's the Arizona Gila monster. It's the cool one. It's the venomous one. It's the one that eats eggs and has armored skin and lives a long time, and it's very large, but not that large, but has these gnarly teeth and is extremely venomous, and it's just a very cool thing. And I'm super glad that we get to work with Dale on these animals because look at that. That's cool. They're cool. Look at that. He's just looking at you. This is the only one I've ever seen in the wild, but so you get my granulated picture because look at it. So awesome. And but I study the genome, so let's look at his genome. So this is basically 10 years of history in one panel. So in 2015, when I started my graduate work, this is the quality of genome. This is the amount of linkage we had where we could estimate what things are attached to what. Not much. Later in my PhD, about 2018, they developed this new technology called linked read sequencing, where we could get slightly larger chunks of the genome, and this is what was used to assemble the Gila monster genome draft that we published last year. Things are moving quick. And then this is our PacBio Hi-Fi assembly, where things are getting much more linked together, but as you can see, they're still scattered, right? So this is like a puzzle piece, and the lights are trying to connect into a complete picture. But once we actually finish the genome, again, all of the light is connected within these boxes that are reasonably difficult to see on the projector. But again, everything's there now. We know everything from this end of the telomere to that. It's all of it is there now, which is cool. And again, I'll just blow it up for you because it's beautiful. So again, we've got our chromosomes, and then we've got this weird thing going on down here we're not going to talk about yet, and then other chromosomes. So again, we know the karyotype, so we know that we're looking for 18 things, or 36 if you're counting the diploid level here. But we know that the sex chromosomes are here, and there's 18 pairs, and these ones are metacentric, and these ones are acrocentric, and those are the things that we knew going into this, which is cool. But again, we have our graph. This may look familiar from earlier in the talk, and we have all 19, so we have 17 pairs plus the Z and the W, and the mitogenome, because that came with it just for free. And so for the rest of the talk, the next 15 minutes is all future directions and just tidbits of fun stuff that we're learning as we go. So we have sort of a series of questions, and I'm just going to touch a little bit on each of them. First things first is, obviously, if we had a very terrible genome, and then a pretty good genome, and then a really good genome, what have we been missing up until now? So if we align our PacBio assembly to our new telomere-to-telomere assembly, we'll see, again, that's pretty good coverage. That's probably like a centromere area. I don't know what that is, but every time you see a color break, it's where there's no coverage, right? So here's some microchromosomes. Always the end is sort of missing. Again, probably a centromere area here, the end of this chromosome. But mostly, we had everything, mostly, right? So it's good. Here's what our 10x assembly looked like, just that we published last year, which leaves some wanting all across the board. But yeah, again, like half of chromosome 2 is like missing, and again, there's a modest improvement, so that's good. But what are we missing? So we have nanopore data, ultra long nanopore. We have PacBio, and so some work being done in the lab by Carmen Webster here at ASU. She's gone through and analyzed basically which regions of the genome are better sequenced by nanopore, which regions of the genome are better sequenced by PacBio. And so if across the line, again, if it's around 0, they're the same. If it goes above the line, this is a nanopore bias. If it goes below the line, it's a PacBio bias. And so we're getting to explore a little bit of what's going on here. And so we took the features from these windows that were significant outliers, and what we find is basically a lot of repetitive stuff. So we have DNA transposons, a couple of genes were in there. Be interesting to see what genes those are. A bunch of lines, signs. This one is just sign question mark. I just kept that one in there. But these are just the ones that have like actual differences between them. And then of course we have the unknown, because in squamates we deal with a lot of unknown for some reason. And so just a cursory look at some of the things that we're now obtaining that we never have seen before in these animals. And of course, where are the sex chromosomes? Because I am who I am. And we can run our little SCINK pipeline, and we can find the W way up here and the Z down here. And one thing that you'll notice, again, is that the W is normally like here, and then the Z is usually like here, and it's all inverted and whatever. So also, if chromosome length is along the x-axis, which it is, and this is the lowest point there, which means that the Z is the smallest chromosome, which of course means that this cannot be the Z, because if it's here, then it can't be here also. But that is the W. Of that, we're really reasonably confident, because that staining would suggest that it's the W. And so if we fix the karyotype a little bit, we'd actually see something more like this. And so this angomorph that we thought had a homomorphic sex chromosome system is actually extremely heteromorphic. So we've taken the karyotype, we've used it to inform our sequencing, and we've taken the sequencing and we've used it to inform the karyotype. And actually, we've talked to the authors of this paper, and they are redoing the karyotype, and they are going to map exactly which chromosome belongs with the W. So that's an exciting new ongoing piece of the project that we're interested in seeing the results of. But as I already alluded to, with the what have we been missing, we need to talk about repetitive elements. And so if you looked at pretty much any squame genome currently available, you'd see something like this, where this is basically a pie chart of the total content of the genome, and each of the colors is some kind of element that could be found. So in this case, red is DNA transposable elements, blue are lines, pink is simple repeats. This gray area is the unknown. So 30% of most squame genomes is unknown, because that's where we're at. And then, of course, we have this big chunk that's non-repeat. And this could be any given genome for a squame, but in this case, it's chromosome 1 from the Gila monster. And so if we take this and we use it as sort of our baseline to look at the sex chromosome, Z specifically. So it's the biggest chromosome in the genome. This is the smallest one. And proportion-wise, it's very similar, right? Like, again, the unknowns are a little less, but there's less chromosome to be had. Relatively more lines, but generally speaking, looks pretty similar. So now if we replace chromosome 1 with the W, we see something a little off, which happens to be that 50% of the chromosome is actually satellite repeats, which would help explain why it's so large. And indeed, even if you align them, we just see these really high identity cassettes of satellite arrays just amplified over and over again, such that the Z chromosome is only 20 megabases, but the W chromosome is over 70. So there's a lot going on there, and we're really excited to dig in a little bit more, especially because it looks like there's been potentially some inversions here on the W that have reoriented genes. And again, there are still regions that align rather well to parts of the Z. Again, here's the pseudoautosomal region that still recombines. And then there are these regions of just nothing, right? Like this is just a cassette. There's no genes. Doesn't even align very well. So a lot of fun stuff to investigate there. And as we're talking about sex chromosomes and what's on them, I want to bring us back to the paper we published just last year talking about dosage in Gila monster. And if you look at this really strong, significant evidence of 1 asterisk between this little bit and that little bit and this little, and it's just, you know, like if you squint, you're like, man, that's really convincing stuff. And I need to point out that there were 60 genes included in this analysis, because that is about the level of how much of the Z we had was 60. Again, just that little chunk of the Z was in our last paper, but there's about 300 genes on the Z. So now we can revisit this question with increased statistical power and perhaps generate something a little bit less squinty when we look at it under the microscope. So a lot of this is revisiting similar questions with new data, but it's always exciting. And then I want to just briefly touch on microchromosomes, because that's, you know, there's 5 macrochromosomes and the rest of them are microchromosomes, and microchromosomes are different than macrochromosomes in a lot of different ways. So if you look at our high C contact map again, these are macrochromosomes. Down here are microchromosomes. And what you'll see is, unlike here in the macros, and maybe you'll see, where there are these hard, distinct boxes, down in the micros, that's not really what you see. You see a lot of fuzzy, with the exception of this black box here. you see a lot of fuzziness going on here. And that's because microchromosomes interact with each other strongly in the nucleus. So they actually form some kind of a structure inside of the nucleolus that is presumed to have some kind of function. We just don't know what it is. And squamates are an extraordinarily great model for this. And you say, wow, again, they're a great model for something? Who would have thought? It's because squamates have lost microchromosomes at least twice independently, one of which is in geckos. So again, we just have this graded series. This is something similar to what you'd see in like a human, but you don't see this really strong bias at the small chromosome level. So obviously this structure is not existing in these taxa that have lost microchromosomes, and so this may give us a little bit of insight into how things are going there. And then telomere evolution. So telomere to telomere, it means we have telomeres now. We didn't have telomeres before, so I'm going to keep saying telomere over and over again. And the telomeric repeat in vertebrates is GGG-TTA. You can reverse complement it, you can do whatever you want. But if we look at our chromosomes towards the end, so these are all the macros, and these are not aligned in any way. These are just the sequences, and they're all color-coded. So yellows are Gs, Ts are green, and then an A is red. And this bottom one here is a telomere. Okay, it's definitely a telomere. Double-checked, triple-checked. This is the telomeric repeat. These three are not telomeres. But they look like telomeres, don't they? Well, those are the centromeres, because apparently, at least some squamates in the microchromosomes have centromeres that are just extended versions of the telomere, which means that when you look at them in like a graph format, towards the end of the chromosome, so all these green tags mean that what it's attached to is a telomere. And so these are all bits of the genome that are being flagged as telomeric, but they're in the center of the chromosome because squamates are weird. Who would have thought of it? But so these are the kind of things that we're interested in looking at, and this is especially strong in the microchromosomes. And I may or may not have a slide later talking about that, but if not, that's fine. Let's move on. And then the last thing, and perhaps one of the more iconic things about the Gila monster, is its venom. And of great interest in the venom is these 4 proteins called exendin 1, exendin 2, 3, and 4. And so one of our projects is, where do these come from? How do they evolve? And so first and foremost, we just took the genes from NCBI and we blasted them to the genome. And we found this cassette on chromosome 2 that contains exendin 4, exendin 2, and exendin 3. And importantly, I must mention that this exendin 3 is not from a heloderma, it's from another species. But it's thought that exendin 4 evolved in the Gila monster to the exclusion of all other beaded lizards or helodermatids. But I'm going to show you 2 screenshots really quick. I need you to pay very close attention, because if you don't, you're going to miss it. So here's a zoom in of the exendin 4 region. And so this is the Gila monster reference sequence. So we have exon 1, 2, and 3. This is, again, exendin 4. I need to emphasize this is exendin 4. Exendin 4 mRNA. Are you ready? Okay, so now we're looking at exendin 3, okay? So this is obviously a very different gene that occurs in a very different location and is it's certainly not the same thing. So so again, these are the kind of insights that we're now getting, in that we thought that exendin 4 was some Gila monster specific duplication. It turns out it probably is not, and it's actually orthologous to exendin 3, which occurs in all the other helodermatids. So that's all cool and interesting, and which also begs the question, if this is actually exendin 3 from beaded lizards, then what is exendin 3 from all other squamates doing over here? And how are these genes even related to each other? So that's some work that's going to be taken over by Danny Jackson in our lab, which we're really excited about. And then as we wrap up here in the last 90 seconds, the logical conclusion to what I've told you today is we should study the Komodo dragon. And so, I know you're like, it's obvious, right? Okay, but we partnered with the Phoenix Zoo, and we now have blood from Jojo. This is not Jojo. This is McLeod, the other one, the male. But they have a female Komodo dragon, and again, Komodo dragons have a ZW, so we're interested in the females. And we are very excited to sequence the Komodo dragon as the other famous venomous lizard and compare what we find in the Komodo dragon to the Gila monster. I'm just going to stop that. And most importantly, in January, JoJo celebrated her 12th birthday, and this QR code will take you to Instagram, and you can watch her little birthday party, and I highly recommend it because it's a joyous occasion. I gave her a box, and she was playing in it. It's very cute. I highly recommend. And if not, Phoenix Zoo, JoJo, 12th birthday, it'll come up. So let's move on. And then again, I want to flash back to one of the very first slides I showed you. So this is the Biology of Lizards conference last year. There was a whole Heloderma day. It's all these really impressive experts in the field, and then me at the bottom. You can see me down there, totally I can't believe they let me sit in the picture. But I want to point out that although I got to meet all these great people, and Dale, I also got to meet this guy next to me. His name's Daniel Ariano Sanchez, and he is what I would consider to be a primary reason that the scorpion of Montagua, which is actually a Heloderma, is still alive on this planet. So Heloderma Charles Bogarty, it's located in the sister group to the Gila monster, and it's very endangered, CITES 1 listed. We are now working with Danielle to sequence their genome as well, so we have another taxa to compare the Gila monster to, and it's very exciting, mostly because this collaboration allowed me to hold one, so yeah. And then I get to sequence it too. That's a whole kit and caboodle there. So boom, real talk. So we are very excited. This is just a couple snapshots of all of the things we're getting ready to do. I have special thanks to many people that either pictures were in the talk or not in the talk, but they all helped in some way, shape, or form to get this far and move forward from here. And with that, I will take the questions.
So recently, I learned from another talk of yours that the sex-determining chromosome in the heteromorphic systems is not always smaller. Do you have to be sure to collect a particular sex? Like, do you need both male and female in order to identify the sex chromosome system? Like, how do you?
That is the most assured way, is to sequence them both, and then you know that if there's a region that occurs in one and not the other, that's probably it. But as we sort of looked at earlier, that's not always the case. And there's actually, although any chromosome can be a sex chromosome, and these things evolve fairly quickly, there does seem to be some predisposition for certain groups to maintain certain types of sex chromosome systems. For instance, in anglomorphs, there's at least 5 different ZW systems just within that. It seems like there's some constraint around being a ZW system, but it's not entirely clear why that would be. But we see that in a lot of groups, where once you're sort of in this mode, you tend to maintain it.
For the Gila monster, what did you collect for those samples?
So our first genome was a male, ZZ, and the one we just sequenced was a female, ZW.
I wanted to ask a little bit more about the telomere-like sequences that you're seeing in the centromeres in some of the microchromosomes, and I'm just curious if you're seeing normal telomeres in those microchromosomes, and then what kind of functions would you, like, if you were to think about why that might be in the centromere, like, what do you think it's doing?
So these guys we're close. There we go. So this centromere motif-ish appears to be in the, well, one, it's all over the W, first and foremost. A lot of the satellite arrays are like weird telomere things. So that's a pin. But all of these blank areas are actually the centromere-telomere construct. So these are all acrocentrics, and so there's going to be a telomere at the end. It's just weird that it's sort of merged with the centromere. And the centromeric repeats are different enough where the data can assemble it fine. It's not like 8 megabases of telomere, like there are differences. And so, but what's interesting is that a lot of people are, they have their PacBio data and they say, we did it, we have telomere to telomere, even though it's not complete. Like, look, we have one telomere here and one there. It actually looks like what they did is they reached the centromere. And PacBio then would stop sequencing once it reaches that limit. And you would think, because it's actually telomeric, centromeric stuff, it looks like a telomere, but they haven't actually reached the telomere. And then one of my pet hypotheses is that this has something to do with that structure inside of the nucleus. It can't be a coincidence that they're like all doing it. Like, I don't know. Maybe it's Gila monster specific, but I doubt it.
So we do have an online question, so let me read that. Can you say any more about what you think might underlie the difference between PacBio and Nanopore in terms of the regions they are differentially assembling?
So aside from sort of what the features are that appear to be in this region, it does seem like it's sort of the classic. And what's interesting here is, again, the orange is the PacBio has sequenced more of it, and the blue is Nanopore has. But the regions in which Nanopore does better are sort of the traditional PacBio kind of stinks at GA-rich regions, and these are enriched for GAs. And satellite arrays are the big one as well. And of course, unknown. Ooh.
Any additional questions? All right. With that, I would once again like to thank Dr. Brendan Pinto.
Thank you all for listening to me gab.